Assalamu alaikum dear aimers. Hope so you all are doing well. Today the topic of our discussion is non-cyclic phosphorylation. So let's start today's topic. Firstly, we will look at the contents what we have to study for today. So here is the content list. Firstly, we will look at the forms of light reactions. Then we'll go through phosphorylation, non-cyclic phosphorylation, definition, properties and process. So basically the main topic is non-cyclic phosphorylation. So firstly, we will see what are the forms of light reactions. So there are two forms of light reactions on the basis of pattern of flow of electrons. Okay, so on the pattern of electron transport or on the pattern of flow of electrons, there are two types of light reactions, or there are two forms of light reactions. First is called as non-cyclic electron flow. Like electrons do not follow a cyclic path, rather they transport into a non-cyclic pattern. And second is the cyclic electron flow, in which electrons flow in a cycle throughout the process. Now the form of a light reaction in which electrons are transported in a non-cyclic pattern, that is called as non-cyclic phosphorylation. And the form of light reaction in which electrons are transported in a cyclic form, that is called as cyclic phosphorylation. You may look here in the diagram that the pattern over here is non-cyclic. You may see that this is a Z shape, okay? So electrons are transported in a Z shape or zigzag pattern. That is why it is called as non-cyclic phosphorylation. While in case of cyclic phosphorylation, you may see that it forms a cycle. Electrons, the pattern of electron transport forms a cycle. That is why it is called as cyclic phosphorylation. Now, what is the meaning of phosphorylation? You see, wherever the word is lation, it means the addition. Like glycosylation means the addition of lipids or proteins to carbohydrates. Okay, so wherever the word is lation, it means that there is addition of something. Similarly, phosphorylation means the addition of phosphate group to any organic compound. That is called as phosphorylation. For example, when phosphate is added to ADP and forms ATP, so this addition of phosphate to an organic compound ADP is called as phosphorylation. Now, what is non-cyclic photophosphorylation? Non-cyclic photophosphorylation means the production of ATP by the light-dependent reactions in a non-cyclic electron flow. We got it that phosphorylation is addition of phosphate. It means it is the production of ATP. Photo, in the presence of light, the ATP is produced. That is what is called as photophosphorylation. And the electron pattern, the flow of electrons, is in a non-cyclic pattern in the whole process. That is what is called as non-cyclic photophosphorylation. So this is the definition of non-cyclic photophosphorylation. Now, where does non-cyclic photophosphorylation occur? As it is a light reaction, so the non-cyclic photophosphorylation occurs in a thylakoid membrane of chloroplast. What are some properties associated with the non-cyclic photophosphorylation? Do remember that this non-cyclic photophosphorylation is a continuous process. It occurs continuously in the plant cell. Okay? Whenever the light is provided, the non-cyclic photophosphorylation will be started in a plant cell. And one more important point, that it is a process which happens in normal routine. Like when plants are in a normal condition, they don't have any deficiency of anything, then the process occurring in a normal routine is of non-cyclic phosphorylation. Okay, so these are two properties associated with the non-cyclic photophosphorylation. Now, what are some important points which you have to remember for the non-cyclic photophosphorylation? Firstly, the reactants of non-cyclic photophosphorylation are same as of the light reactions, because it is a form of light reaction which is light energy and water. And what are the products which will be formed in a non-cyclic photophosphorylation? That is ATP, NADPH and oxygen. Oxygen is actually a byproduct. The main products are ATP and NADPH. ATP has chemical energy stored in it, while NADPH provides the reducing power. Now, what is the flow pattern of electrons? We have discussed it earlier that flow pattern of electrons is non-cyclic or zigzag shaped, or you may call it as Z pattern shaped. Okay. Now, what is the type of this photosynthesis? This photosynthesis type is called as oxygenic photosynthesis, because oxygen is produced in a non-cyclic photophosphorylation. And how many photosystems are involved in the non-cyclic photophosphorylation? Both of the photosystems, photosystem I and photosystem II, they both are involved in the process of non-cyclic photophosphorylation. So let's see what are the steps involved in a non-cyclic photophosphorylation. So there are many steps involved in it. First is light is absorbed by photosystem II. Second step is the photolysis. Third step is in the third step we will study in detail the electron transport chain of photosystem I. I just gave you its overview in the last lecture, but in this lecture we will study its detail. Next point is ATP synthesis, then light is absorbed by photosystem 2, and then we will study the electron transport chain of photosystem 2, and then NADPH synthesis. I have committed a mistake over here that here we will study the electron transport chain of photosystem 2, and here we will study the electron transport chain of photosystem 1, because photosystem 2 functions first while photosystem 1 functions later. So first step is the light absorption by photosystem 2. What happens that antenna pigment molecules, chlorophyll A, chlorophyll B and carotenoids, all these molecules absorb light energy and they transfer light energy to the chlorophyll A of a reaction center of photosystem 2. Okay, you can see in the diagram that photon falls on the molecules of antenna pigments. Antenna pigments absorb light energy, and this green part is of chlorophyll A of reaction center of photosystem 2. Photosystem 2 is performing its function first, and then electrons are excited. So first step is that light is absorbed by the chlorophyll A, which can absorb maximum wavelength of 680 nanometer of photosystem 2. Then what happens that electrons, by absorbing the light energy, get excited to a higher energy level. Now look here that as the electrons have been removed from the photosystem 2, so we say that the chlorophyll A of the photosystem is now oxidized because electrons have been removed from the chlorophyll A of reaction center of photosystem 2. So as electrons have been removed, you may look here that this is chlorophyll. Two electrons basically are removed from the molecule of chlorophyll, and this chlorophyll undergoes oxidation. The removal of electrons is oxidation, and as chlorophyll becomes deficient in electrons, it gets positive charge because electrons have been removed. So there is electron and proton imbalance. The charge is positive charge is greater than the negative charge. Now this chlorophyll, which has positive charge, which has lost the electrons, is called as oxidizing agent. Oxidizing agent means the agent which will reduce itself and oxidize other. Now this chlorophyll plus needs the electron. It will accept the electron. That is why it will undergo reduction. Okay, so this is an oxidizing agent. This chlorophyll undergoes oxidation. Electrons have been removed. When electrons are removed, positive charge is created. This positive charge will be removed by the acceptance of electrons, and whenever the electrons are accepted, it is called as reduction, and the substance which undergoes reduction is called as oxidizing agent. Got it? Now the excited electrons, these two electrons which have been removed from the chlorophyll A of reaction center, will be accepted by the primary electron acceptor. This primary electron acceptor is protein in nature. Now look here in the video that photon falls. This is reaction center P680, chlorophyll A. Electrons get excited and are trapped by the primary electron acceptor. Got it? Now what is the second step? The second step is of photolysis. Now wherever the word is lysis, it means breakdown. Okay? Lation means addition and lysis means breakdown. Photo means in the presence of light. So it is the breakdown of something in the presence of light, and that something is water molecule. So splitting of water molecule in the presence of light is called as photolysis. Now photolysis is done with the help of an enzyme. This enzyme is attached to the inner side of the thylakoid membrane, and this enzymatic complex helps in the splitting of water. And where does photolysis take place? Do remember this. This is the most important point, that photolysis takes place in the lumen of the thylakoid. It is the only process which does not take place in thylakoid membrane. It is the process which will take place in the lumen of the thylakoid. Now do you know what is the lumen of thylakoid? This is thylakoid structure over here. Okay, this whole stack is grana, and one structure is of thylakoid. Now the inner side of the thylakoid, the matrix of the thylakoid, you may see here the gray part. It has been dissected from here. So this gray part is called as thylakoid lumen, and it is the place where water molecule will break down in the presence of light. So why does this water molecule break down? Water molecule breaks down because when electrons are excited from the chlorophyll A, the positive charge is created on chlorophyll A. You call this positive charge as a hole. A hole is created in the chlorophyll A of the reaction center of photosystem 2. Now when water molecule breaks down in the presence of light, its electrons are removed from the water, and those electrons fill the space in the chlorophyll A molecule. So the chlorophyll plus accepts the electron and undergoes reduction. And this is the process which produces oxygen as a byproduct. In the earlier lectures, we say that light reactions produce oxygen as a byproduct, and we get that oxygen as an atmospheric oxygen. Okay, so this oxygen which is produced in a light reaction is actually produced in the photolysis step of the light reactions. Now look here how the electrons are produced and how this water molecule splits with the help of water. So as the light energy falls on this water molecule, the water molecule splits into two components. First is hydrogen and second is half oxygen, because it has one oxygen over here. So 1 by 2 O2 means half molecule of this O2, half molecule of O2 will be single O. This molecule of hydrogen further splits into its components with the help of light energy. One hydrogen has one proton and one electron. Two hydrogens will have two protons and two electrons. Okay, now these two electrons fill the hole in the chlorophyll of reaction center from which two electrons were excited to the higher energy level and were accepted by the primary electron acceptor. And this whole process takes place in the lumen of the thylakoid. Now look here in the video that this is water splitting enzyme attached to the thylakoid membrane, and then water splits into two hydrogens and half oxygen. The electrons from the water fill the hole in the chlorophyll of the reaction center. So this is the process of photolysis, which produces oxygen as a byproduct. Now we will look at the third step. We will look at the electron transport chain of photosystem II. So electron transport chain of photosystem II has three electron carriers. First is plastoquinone, and we represent it as PQ. Now, what are the properties of plastoquinone? Plastoquinone is isoprenoid in nature. You may have studied in the topic of lipids what are isoprenoids. These are basically the derivatives of lipids. So, plastoquinone is isoprenoid in nature. Second is, it is at high energy level. You may look here that this is energy level arrow, and this plastoquinone is on highest energy level. Then the second electron carrier is the cytochrome complex. Cyto means cell and chrome means color, because it provides color to the plant cell. Okay, so cytochrome complex, it is 2 in number. Do remember that there are two cytochromes in the photosystem II, and these two cytochromes are B6 and F. Only MDCAT students will remember it, but you have to remember that these are 2 in number. Second is, these are iron containing proteins. Okay, so this is protein in nature and this is iron isoprenoid in nature. There is one more point mentioned in the topic of respiration in your book, but I have mentioned it here, that cytochromes contain heme as a prosthetic group. As hemoglobin contains heme, similarly the cytochrome also contains heme. This heme is same as that of the hemoglobin. And what is prosthetic group? You know that when the non-protein part is attached to the protein part, it is called as prosthetic group. So the non-protein part is heme or iron, and it is attached with the protein part, which is cytochrome. So we call it as prosthetic group. Now third electron carrier is plastocyanin. Do remember that plastocyanin is a copper containing protein. Okay, as it is a copper containing, so it is blue in color. So you will remember it with the help of cyanin, C and copper containing. This is very important for the MDCAT MCQs. So you have to remember that which electron carrier has which type of nature. Plastoquinone isoprenoid, cytochrome iron containing protein, and plastocyanin is also a copper containing protein. Now we will look at how the ATP is synthesized. Okay, so this is photosystem II. Okay, and here is you may look here, this is chlorophyll and the primary electron acceptor, then plastoquinone, cytochrome complex, and plastocyanin. Plastoquinone is at highest energy level, and then cytochrome complex, and then plastocyanin is on lowest energy level. Then now look at this animation over here. The photon falls on it, reaction center P680, electron is excited to primary electron acceptor, and now this electron goes on to a series of electron carriers. The whole is filled by the molecule of water over here in photolysis. Now it goes to plastoquinone, then cytochrome complex, and then plastocyanin. It is high energy level, and it is plastocyanin is on low energy level. So when electrons transfer from high energy level to the low energy level, then they release energy, and that energy is used by thylakoid membrane to synthesize ATP.